Yesterday, we told you about Kenya's efforts to stem HIV infections in the country. Today, we return to the Kenya Medical Research Institute to examine the process of testing and extracting the HIV virus from blood samples.
HIV is one of the leading causes of death right now in Africa. Not only in Africa, but the whole world. But Africa bears the biggest brunt of the issue of HIV/AIDS.
This is Dr. Elijah Songok, the Principal Research Officer at the Kenya Medical Research Institute's HIV/AIDS unit. Dr. Songok takes us on a tour of the laboratory where critical research work on HIV takes place. He says since the mid-1990s, research has been a priority as one of the strategies for fighting HIV in Kenya.
These tubes contain what we call a DBS, a dried blood spot, which has been obtained from the kids. So this is for the early infant diagnosis. So you know, as a small kid, you cannot bleed and take a whole blood samples. The kid is tiny. So you have to take just a small prick from the kid's heel or from the kid's arm and then put it on small filter paper. That filter paper is called a DBS, or the dried blood spot. So you bring that dried blood spot and then you'll be able to put into those tubes and then do some extraction. So this is the tubes that, first of all, we add a buffer, and then we start the extraction process to be able to extract. If there is any of the DNA for the virus, we'll be able to start now extracting from this tube. And then we move it right away towards the machines to do the archival extraction, and then towards the PCR for the amplification. And then we could be able to detect, using the issue like that computer, that this particular virus or this particular kid has got this particular virus.
The laboratory receives up to 100 samples a day from hospitals across the country. To expedite the process, the laboratory uses an extraction machine.
Again, this makes the work much more simpler because of its automatedness. Yeah, so handling those 500 samples a week, if it was personnel, the number of people we'll be having here will be so much. And again, easy to make mistakes. So when you have an automated machine like this makes much more work easier. Yes.
It takes 5 days to run the tests, determine the status of a particular sample, and to send samples back to the clinics where they originated.
So this is how the samples come in from the clinic. So they come on a DBS. So you have right now this filter paper. You have around 5 blood spots from each kid. So just stick. Wrap it up and then send it. The good part of this is that it does not need refrigeration. It's easier to send from even a very rural area. So just send it by post and arrives here. This is very simple indeed, just like a letter, putting it in a post office and sending it, or a courier, just bring it over.
Once you get the results,
yes,
you get the results from all the samples.
Yes.
Apart from sending them back,
yes,
with the results, whatever it is, whether positive or negative,
yes,
are you using the samples for perhaps to isolate the different strains of the HIV virus and try to do your research and studies on that?
Yes, first of all, there are various types of samples. So there are samples which come specifically for diagnosis. So those ones we do the test, diagnose, and then send the results. We can be able to use those same samples later on for our research. But this we have to undergo another special, we have to ask for permission, ethical clearance. And then we have to get the guardians or the mothers, this is for these kids, to be able to consent that please, you can be able to use this for research purposes. So that would be another step.
The Kenya Medical Research Institute conducts research on other diseases, and next week we'll take you to the malaria laboratories.